I very basic question, but I'm not sure. It turns out that if you look at the literature as usual, the basic questions, you hardly find the answer because this field gets so big and so specialized that people don't think about fundamental problem anymore. So it is a kind of child's question, if you want, very basic question. And okay. Does the host want anything else from me? So Travis is now in Texas, Adrien is now in Dresden. Christopher Alexanderson is in Oxford, student, Georgia and Prama and Pavan also. Andrew is now in Stanford. And if we can see the name below, collaboration with the group of Thomas Knowles and Georg Meisel in Cambridge, who work on the aggregation process experimentally. Long-time collaboration with Ellen Kuhn in Stanford. And the last part of the talk, and I'm not sure I have time to explain about brain dynamics and brain wave, that's done with Chris Bick and Willem de Haan in Amsterdam. Okay, so let me start. Let me tell you just a little bit about dementia. I know that people here are working on that, but just to set the basis here, let me tell you about William Utter Mullen, who was born in 1933 in the US. He moved to the UK in his 30s to go to the Ruskin School of Art in Oxford and became quite a well-known painter with very modern style. But in his 50s, in his late 50s, he started developing some worrying sign. He couldn't quite know to tie, things like he'd do every day, do the home finance, he would forget things, he was depressed. So eventually he went to his doctor, and 4 years after the his symptoms showed, he was diagnosed with what we call probable dementia. He was a very good painter, and his wife was an art critic. And so they decided to keep a diary of some sort of the progression of the disease. And I think that's one of the most moving testimony of that we have. Here is him, a young man, before dementia, and probably at the peak of his career, he already had sign of dementia by the time. But you see the use of very bright color. He was very successful in his career. But then, very rapidly after the diagnosis was made, you start seeing a systematic degradation of his ability to paint and the sense of self he had also. This is 97, 98. And you see that right away it becomes much more flat, the use of color is a little tentative. It becomes much more abstract in a way. I mean, I still like his later self-portrait actually. But then that's the very last one we have, that it really shows the inability both to visualize, but also to be able to control even the muscles of the hands and all he could do before. And this type of progression is very systematic, unlike other diseases, unlike cancer, unlike heart disease, unlike many viral diseases. Neurodegenerative disease are what's called homogeneous disease. It means they essentially present themselves in the same way systematically. The progression is very well known and classified now to the work of the Brack and Brack, husband and wife, who have looked at pathology of diseased brain at different stage of the disease, in the evolution of the disease. And what you see here at the little part here, the dark part, is the one that are coded, or the one that are marked for protein called tau protein, which is believed to be the main culprit in the symptoms, not so much both in the progression of the disease, but in the symptoms that are seen, and that start early on in the staging of the brain, in the lower part there, associated with the entorhinal cortex. And then the protein the toxic proteins spread within the brain, and at a further stage you have systematic evolution of that front, and you see that after a while in all cortical area. And this is associated with, you see that's another view of the brain. This is more coded now. You'll see that start at the entorhinal cortex and go within this early brain region, and then started moving around the brain and attacking every single part of it. And this type of progression is associated with tau. The hallmark of Alzheimer's disease, which is Alzheimer is the main form of dementia, maybe 60 to 70% of all dementia are actually Alzheimer's disease. I will talk mostly about Alzheimer, but the other ones, the other neurodegenerative disease that are also proteinopathy caused by proteins, follow the same type of mechanism. Everything is a little bit different. Alzheimer's disease was characterized by Alzheimer with the presence of these two proteins, these two aggregate of protein, amyloid beta, amyloid beta deposit, and tau. Amyloid beta is a protein that is extracellular, it's intramembrane, but it's the deposit at extracellular. Nobody knows actually what is the function of healthy amyloid beta, which is quite remarkable when you think about it. It's clearly associated with the disease. Tau protein are smaller protein, the monomers are small protein that are intracellular. They're there to stabilize microtubule. That's what gives the rigidity of the axon and the integrity of the axon. Alpha-synuclein, which is also associated with Parkinson, also has the same type of function. But these can go wrong and create this large aggregate, as we're going to see, that systematically invade the brain. And at the cognitive level, so you have this evolution in space and time of the disease, and at the cognitive level, it's also attacked different parts of the brain early on in the hippocampus that's associated with memory retrieval. So that's a problem with amnesia or anomia, the inability to recall certain terms or certain name. And then later in stage, you have agnosia, the inability to recognize face and other things. Apraxia, problem with small movements. Aphasia, problem with either understanding or formulating sentences and speech. And then it goes on later in the disease, much more severe, anosognosia, which is the inability to recognize your own disease. And that's why so much problem with dementia patients. They don't really understand that they have a disease. Long bouts of apathy, can last for hours, and a very high level of anxiety. And it's a very cruel disease because it attacks the sense of self. People really lose their own identity, and it's cruel for the person, but also everybody involved. And since medicine has been so successful over the last 20, 50 years, and we know to treat a lot of cancer, we know to treat heart disease, some of them, now to the point where dementia has become in the UK the first cause of death, actually. One out of 9 people over age 80 has some form of dementia or Alzheimer, mostly. So, as I said, other neurodegenerative disease follow the same type of evolution. Again, for Parkinson, it starts in a different region, close to the brain stem, and also systematically invade the brain. It's associated with alpha-synuclein. Amyotrophic lateral sclerosis, ALS, also associated with, start in the motor cortex and then propagate to the brain. And you see there's something bizarre with ALS, is that mostly the back part of the brain seems to be protected from the disease. It's associated with another protein, TPD43. So all these have the same type of progression, and as I said, very homogeneous in the way they present themselves. So the physiological effect of that, so that's the level of protein, but what is, I said they were toxic protein, and they're toxic because they start killing cells around them, both amyloid beta, but mostly tau. Tau is closely associated with brain atrophy. So cells die and they're systematically removed. This is 2 brain, one which was healthy at the age of death, around 80, and the other one with Alzheimer. And you clearly see the difference. And the first time I went for autopsy of a brain, the person in charge, the doctor says, "Oh, this is an Alzheimer patient," you know, directly looking at the brain, and you see it is kind of walnut dry aspect of the brain, very different than the full brain. And if you look in a little bit more detail, you'll see that there's very marked difference between the two. Atrophy takes place. You have larger ventricle, and that's a mechanical effect because as the material is removed, it's somewhat the ventricle enlarge and a pull. You have an opening of this sulci, which has this valley of the brain, you know, this is very intricate. You know, you have room for that. You have a thinning of the cortex. Cortex is typically, for most mammals and for humans, between 2.5 and 4 millimeters. And you can see a very sharp early decline of the cortex width. And you have a shrinking of the hippocampi, very marked, because that's where the disease starts. So in that region, so that's where it's mostly observed. You know, people say, well, all brain age, all people always get a little lost and all that. Dementia is just a weak form. That was something that people thought maybe a century ago, that was a normal evolution. There is healthy aging, volume change. Yes, we lose a little bit more, there's so much neurons, but nothing like what you see in the atrophy of Alzheimer. It has nothing to do. And healthy aging also means that you are a little slower, that because the connections are not quite as fast, and that you also lose some mass, but the mass that you lose might not be that important. And maybe you lose a little bit of some memories, but actually in most long-term memory, there is no difference. So this is really completely different than, you know, fast aging or something like that. So if you look at the literature, it's huge literature, the entire journal dedicated just to Alzheimer and other of these diseases. One of the basic tenets in many reviews is what's called Jack's curve, according to Clifford Jack, who is one prominent doctor. And the Jack curves are the way the medics see the evolution of the disease. So here in this plot, you have different curves. There is no unit on any of these. There is time and progression. But let's look at the first curve and what you see here. So if you see here, this is the curve for, let's say, the amyloid beta. So the increase in the load of the toxic form of amyloid beta. And at a certain point, to some scan, you are able to detect it. And that's the line here, the detection threshold. And for the longest time, and still now, people associate the disease with amyloid beta, and mostly because it's the first one that's observed in scanning, right? And after that, you're able to manage to discover that you have this other protein, tau, in the CSF that accumulates, and that you see the presence. Then MRI, the atrophy kicks a little bit later, because it's an accumulation of damage, essentially. And then you start seeing in MRI the trace of that. And now MRI is one of the main biomarker to diagnose the disease. There is a change in the 80s before the 84, the diagnosis was probable Alzheimer, and it was only called Alzheimer after that, when you actually look at the pathology of this protein. But now we have sufficient understanding, and if you have an MRI plus a certain number of cognitive tests, MRI that show atrophy, that's enough for a lot of the diagnosis.
So excuse me,
yes.
Can I ask the question?
Sure.
So that means that this is the very first time that you see a reduction or shrinking
Yeah.
of the volume?
Yeah. Well, now MRI, you know, as I say, there is no scale or here, so what does that mean extra? And then you have supposedly all the cognitive functions starting here. So that's for medics, that's what some model says. So we said, that's great, you know, there is a model there. So you go back to the paper, you look at the, you know, the supplementary material, and soon enough you realize that the idea of a model is drawing a line on a piece of paper or Adobe and putting in the paper and having, you know, 5,000 citations for the model, okay, which is perfectly way. It's a way to synthesize the subject, but it's a very different way of what we would call a model. If we try to write a paper to Sci and we say, here I have a model, you know, look at this nice curve, I don't think it would go very well, right? So, you know, said, okay, now can we actually understand that? Can we understand all these difference of progression and all that? So the last piece is about cognitive function. So I don't know if I have time. So I time my talk, hopefully, so that the first part is completed. And if there is extra time, we can talk about oscillation dynamics. And that's a very more recent part of our research, where we say, okay, if we know what happens to the brain, or we know the spreading, we know the damage, how does that affect actually the way the brain actually works? Because the brain is all about cognitive function. It's not just a physical object. It's a physical object of its own interest, but it's not a mechanical structure that supports loads or anything. Its function is cognitive. So one way to test that is by looking at brain wave. And so if you put a net like that, either to EEG or MEG, you can record different brain activity. And in particular, you have these meditative activity here that are characterized with alpha waves that you see in the chart. And depending on the level of activity, you know, you're probably very meditative. So if I put the net on you, you know, you either be on the sleeping phase or the meditative phase, or looking at it very alert, so you'll be on the top of the chart. But you can actually record that, and you can do it systematically. It's very cheap compared to any other scan, which is just a net and electrodes and all that. And what is known is that there is a change in brain activity, which is what we expect. There is a lot of change in cognitive response. There is a change in brain activity. And one of the things we understand the best is that within the alpha wave, there are two types of change. First, there is a change in amplitude, that there is first an increase, hyperactivity, followed by hypoactivity. So as the disease develops, your first part of your brains are actually more active. Overall, these waves are higher amplitude, but there is also a shift of frequency in them. So this is the hallmark of the two. Initial followed by hypoactivity and a shift to lower frequencies. So long introduction to tell you what were the questions that we had for this type of problem. So one of the first things we wanted to understand was this spatio-temporal progression. Well, we come from the field of applied mathematics, where spatio-temporal patterns is the kind of things if we see, we try to explain where it comes from. The second one was others. If we know that, how does that relate to the accumulation of damage and atrophy? And finally, can we relate that back to the biomarkers, understand the evolution of different zones and all that, and this progression and the functional decline? And you see that in terms of data, it is a different community, people looking at different things, because the modality of the data is very different. Here you have CSF and PET data and or amyloid beta. You have MRI and FDG PET, which is about metabolism. And then here you have EEG and MEG. So very different type. But really the idea is to try to relate these different aspects of the disease. So let's talk about model. So about now, almost 10 years ago, there was a big progress, different group, in particular Walker and Euchre, came and says, you know, all these disease, they're really characterized by this typical accumulation of proteins, and they call that mechanism prion-like. It's just like prion disease. Prion disease is a proteinopathy. It's much faster and all that, but it's also due to the accumulation of proteins within the brain, like Matco disease, for instance. SATs, neurodegenerative disease, they have the same hallmark. And the idea is that you start with, let's say, a healthy protein, like tau protein, and then some event can take place, a misfolding, and they become toxic. And become toxic in one sense is that when they meet another healthy protein, they can template the other protein and force it also to change the configuration and stick to it. Okay. One simple way to think about it is to think about a zombie movie. You know, you have one guy, so all of a sudden you have a zombie appearing here, and the zombie goes around and finds another healthy person and bites them or whatever. And then they form a little group of two zombies, and then they keep on a rampage. And they keep growing, form larger groups. Sometimes the group fragments and go explore different parts of the little village. and then they form larger and larger group, and you know the movie ends. It doesn't end well. The whole city is over until it forms these large aggregate neurofibrillary tangles. So it's really a process of aggregation and fragmentation. So the prion-like mechanism has two parts, which is the production of these fibers, and the other one is the transport of these fibers. And we'll talk about transport in a minute. So to build a model out of that, you can think of protein of different aggregate of different size. So if Ci is the aggregate of size I, so I equals 1 monomer, I equals 2 dimer, I equals 3 trimers, and all that, and look at all the possible rate at which they can transform. If the monomer meets another monomer, it can become a dimer, and so on, and start building like that. And we know to do that thanks to people like Smolikovsky, who in 1916 wrote a paper about aggregation and fragmentation, mostly for models of clouds, or clouds start forming with little particles and things like that. But there is a nice literature on that, a lot related to biochemistry. And you can write rate equation for this type of process. We tell you that the change of little Ci, the concentration of the aggregate of size I, as a transport process, let's talk about that later. It has a production term, because some of it might be produced by normal cellular mechanisms. So for the normal healthy monomer are constantly being produced because they're needed for homeostasis. It has a clearance term because there is a natural process of removing these particles. There's a nucleation that tells you the rate at which two come together and form a big one, aggregation, and fragmentation. And so there are different ways you can write this. I'm not going to go into the detail. It's very interesting mathematically. They have a beautiful structure. And if you see, it is a very, in principle, infinite dimensional system of nonlinear partial differential equation. Even if you remove the transport term, there are already fascinating problems, open problems, just to try to understand the asymptotic nature of the solution of these equations. We're not going to do that. We've done that also looking at this and try to extract this property. But here, I just want to mention that if you study this equation, you can write an equation for the moment. That's what's typically done. The total number of particles and the total mass of particles. And it turns out that under mild assumption, this moment equation can be decoupled from all the other moments, and then you can study these. And when you study that from a point of view of dynamical system, you can look at it as bifurcation with respect to the clearance. If there is not enough clearance, there is an accumulation. So there is a critical value of the clearance at which, below which you are, well, after which you are healthy and below which you become unhealthy. There's not enough clearance. And if you do that, you can reduce this equation and show that they actually reduce to a much simpler equation for the total mass of toxic protein, which is of the form of anisotropic Fisher KPP equation. So it's a nonlinear parabolic equation, which is very well known. It's one of the basic equation from mathematical biology. And you see why, because it naturally appears a universal normal form of more complicated process. And it has a transport term again here. And we're going to talk about that in a second. And it has just the production term, alpha C, and a saturation term that says that, well, you can only go from C equals 0 to C equals 1. Now, the transport is interesting because the second part of the prion-like hypothesis is that the transport is mostly happening within this axonal fiber, either for amyloid beta because diffusion is faster right outside an axon, and that's why you can get by diffusion tensor imaging, which is an MRI technique, or because the toxic protein are actually within the axon. And so you have fast propagation within the axon and slow propagation outside. You can have propagation outside by exocytosis, and that produce particles that go into the extracellular matrix, and then that endocytosis would be taken up by other part of the system. So it has a fast propagation in that direction and a slow propagation away from that. And here are actually experiments that shows that here, the blue particles that you see here a little prion-like particle, and you can look at the diffusion, the different anisotropic diffusion for this system. Now, of course, we know in one dimension, it's something that was already known by Fisher and Kolmogorov, that when you start with any initial positive data, as small as U1, like this one, on an infinite domain, you would reach saturation locally and then propagate as a wave. And since the system is simple enough, it's just a pull front, and that you get the velocity is related to the parameter of the system. So that's something we know in one dimension. That is, it's a very classic, almost 100 years old type of problem. And there is a nice theorem, the hair-trigger theorem, that says that any initial data, as small as U1, will always lead to solution like that. But we want to port that on the entire brain and see what are the consequences of that. So we have the equation. We have the boundary condition, which says no fixed flux. We have an initial source that I'm going to talk about initial data in a second. All we need is a brain, right? And so for the brain, we put Alain here, Alain Poole, it was at Stanford. We put her in a little scanner. And Mehmet Turk, who's now University of Washington, did the MRI of both of our brain. This one is actually much more interesting than a classical MRI because you don't really see it, but it has a little pillow, and the pillow also oscillates at a given frequency. And so you take very quick MRI in sequence, and then you see the motion of the brain in the process, and from that you can compute the elasticity of the brain. So it's MRI and MRE, magnetoresonant elastography, that you can get. Unfortunately, my data was corrupted, so we only have Alain's brain. So here's the travel size Alain brain. So I have the real size one, but when you travel, it's easier. So we have, this is what the 3D mesh, you see the white matter, the gray matter, the skull, we have the cerebellum and all that. You also need information about axonal direction to get this anisotropy of transport. This is actually the DTI from Johannes Weikenmeier, who was a postdoc and now is at Stevens Institute of Technology. And so we have at each point a vector field that gives us the direction of the principal direction of diffusion. So now, if you start in a region, for Alzheimer, we know that it starts in that region of the entorhinal cortex. You start in that region and you let it evolve. And this is what you get, essentially. So one is at mid-stage, and it's a movie that shows what's inside the brain, and the other one is the external view of the brain. And so you do have progression like you expect. This is a finite element simulation. And now you can do something interesting. You can compare. Let's say, okay, suppose I take exactly the same process with all this prion-like disease. So amyloid beta for Alzheimer, tau for Alzheimer, alpha-synuclein, and TPD43, but I just start a different place. I don't change anything else, just start a different initial condition. What would I get with this simple model? And what you get is actually striking, is that just with this over simple model, you actually get most of the qualitative behavior of the invasion of the brain correctly. It's mostly follow this idea of production, saturation, and transport along the fiber. Very different that if the axonal fibers are very important, the anisotropy is important to get this. And you also see here, we're going to discuss that in a second, I told you the visual cortex is paired in TPD43, and here you don't have, because it's an homogeneous system and there is nothing to stop from that happening. So there's plenty of things to understand. Now, you can relate that to a damage model that says that you have a damage rate equation. The damage accumulates as a function of the protein level. So you add another equation, locally an equation, and link that to the mechanics of the system. So what you do, you run the system, you have the toxic protein, and then depending on the level of this damage, you shrink, you remove mass, you have atrophy of the system. And so this is, we started with, on a slide, this is 2D slide, we started, this is a brain year 1, and we evolve it from year 1 to year 3. And the colors here are not the level of protein, they're just the von Mises stress. So it's a real mechanics computation. And since Alain and I have been playing with the mechanics of biological growth for many years, it was a natural thing for us to look at. And you can really understand now all these features of opening of the sulci, enlargement of the ventricle, and so on, that naturally follow this type of atrophy process. There is an interesting feedback that is not in the system, where you really have to look clearly at the data. We are at a level where we're very qualitative. More recently, we realized that there is a feedback with the fact that since you lose a certain amount of volume, then the data that you have has to be corrected by the concentration because you lose volume. So the concentration actually change. And some of the data shows for some of these markers in tau PET show a decline in tau PET for late aging. And now we have an explanation for that, that it's actually due to that change is due to atrophy in the data. Okay, so it was nice to see that we can capture some of the feature. And so we really asked the next question was, how do we really understand what's going on? What is the simplest mechanism, the fundamental mechanism that makes this thing happen? All we have, as I said, is a production saturation mechanism on an anisotropic grid. And so the hypothesis was it's mostly driven by the topology of this connection within the brain. And so we decided to go a little bit further and reduce that to network. And so the idea now is to replace the continuum on which we had the evolution of this system by a discrete system. Essentially, you find region of interest, and these are very well known. It's in neuroscience. There is a whole literature on this type of thing. They're different, what they call atlas. We isolate region, could be anywhere between 80 to 1,000 or more region, or million or 30 million. And you can go from one to the other one. And so the idea is you identify this region within the brain, and typically they correspond to certain type of well-defined anatomical feature of the brain. And you look at all the connection between these two regions, right? You look at all the connection, and then you replace these by a graph. You say, no, I'm going to forget about the continuum version. I'm going to replace these by a graph of the system. And I'm going to weight a graph, so a graph has nodes. Two nodes, I and J, will be related by the number of connection between the two and inversely by the length. So we don't know how inversely by the length it could be. If it doesn't depend on the length, it would be 0. If it's like a ballistic transport, we know that the reduction of this equation on ballistic transport, the scaling would be Q equals 1. If it's diffusive, Q equals 2. Turns out that it makes almost no difference in any of the computation we made, and we couldn't find any rationale for choosing one or the other. You can argue plenty of different ones. Turns out that in the last part that we'll talk, when we look at the data, it turns out that Q equals 0 is actually the best choice in order to fit with the data, that it really doesn't matter the length because of the type of process that we have. Okay, but it's a minor point that but the important point is that the very important point is that it's weighted by the number of connection. And the idea is that more connection, bigger pipe, more transport between two different parts of the brain, right? So this is what it looked like when you average about 400 brains from the Human Connectome project. And what you see is the weighted adjacency matrix. And it's quite nice. It's this is 1,000 nodes, 1,016 nodes, 1,015. And what you see is very nice. This is a typical brain feature. And it's one of these nice things that I would say anybody who teach linear algebra should show that to the student, because when in linear algebra you show a matrix and you learn something about your own brain, right? This is a case. If you look at that right away, you see that the right hemisphere is mostly connected to right hemisphere, the left hemisphere is really, and that's well known, it's the division between the two parts of the brain. And then you have this low connection right here, corpus callosum, that connects the left side and the right hand side. Also tells you that it's mostly along the diagonal, And it's written in a way that, of course, if you neighbor close in space, you also neighbor in nodes. And that tells you that you're very highly connected to regions that are close to you and poorly connected away from you, except you have this long-range connection also. You see here, away from the diagonal, you have this one, which makes it to like a type of small world network. And all that idea about small world network and use for the brain came from that. And now this type of method for network is becoming a complete classical, well-appreciated tool in neuroscience. People do routinely scan, look at the network, and look at network metrics on that, average lengths and things like that, and try to correlate that with some properties of the brain. It's becoming one of the main tools. And that really is thanks to two things: the development of fMRI starting in the 80s, but really developed for the brain by the turn of the millennium, and the development of applied mathematics theory of network. That the theory of network was understood and developed by people like Strogatz, Danny Bassett, and other ones. But of course, like everything, when mathematics becomes adopted in any other field, it's not mathematics anymore. They're just the same. That's part of our field, which is fine. Okay, but what we're interested in is not so much this metric that you can look, but the transport of this, the spreading of the connectome. And we know to do that, there is a natural way, certainly for a regular grid, to discretize a diffusion operator. And the natural extension for that on the connectome is to look at the weighted graph Laplacian, which is just the weighted matrix with a minus sign. And here is the sum of the weights, okay, which is the case when it's not weighted, it's just the degree of the nodes. It has very nice property. It's also a central object of operator within graph theory. There's a lot of very interesting mathematics using T's as a main object in graph theory, and naturally also appears for transport process. So now we can replace the Fisher KPP on the brain by a much simpler one, which is network Fisher KPP, which has n node. It has the transport process and locally increase in saturation. Okay, so let's see what we can get out of that. This is a type of spreading that you can get. You just start. We start at the node here, the entorhinal cortex, goes to the hippocampi, goes in the frontal, and then visual. So you have a systematic invasion now, one front after another. You start, and that's occur when alpha production is much faster than diffusion term. If the diffusion is faster than the production, then essentially you have a very fast diffusion and everything grows at the same time. It's much more homogeneous, which is what we think happened for amyloid beta. Amyloid beta is and I'll show you that the data confirms that, is very much a process where everything goes at the same time within the brain. So what's nice is that despite the fact that it's an n-dimensional nonlinear system, you can develop some nice new asymptotic methods to capture this solution. This is only for people who like applied math. When you talk about that, you go to a medics or you go to somebody in any other field and says, look, I've got an exact solution, an approximate solution, match perfectly the numerics. I spent 6 months doing this. Well, you had the solution before. What is it? Why are you doing that? I really don't understand. So, what pleases us is not necessarily what other people see as a progress. But look, these are great. These approximate solutions are almost a perfect match. Now, here I've only shown you a few curves because I've coupled all the regions that are associated with this BRAC region that have been associated with the evolution of the disease. So BRAC1 are all the collection of nodes that are the BRAC1 region, BRAC2, and so on. So that means that if suppose that the detection threshold is here, you look here, you first see the disease in BRAC stage 1, 2, 3, 4, 5, and 6. And you see that even with this simple model, all these BRAC regions are ordered exactly like you expect, which I think is fascinating, just as a basic point that, 5 lines of mathematical model and 10 lines of MATLAB code give you the evolution region by region of the disease in what, we've been told for years is the most complicated, untreatable disease. For me, it's big part of understanding what's going on in the system, really the basic fundamental understanding. What you learn if you go in a little bit more detail is that there is a dual process. The primary infection is really a topological property, because from the system, it's directly at connected nodes. If you have one node, you know that the primary infection will be all the connected nodes. You can actually show that. And then you have a secondary infection when the secondary nodes are infected. Now there are multiple paths in which they can invade other parts of the brain. And then it's a balance between the geometry, so are they connected, and the physics. How fast is the connection and how fast is the production at different places. So that was great, and that was a few years back. And you think you understand, but then you send me papers and they say, well, you don't have data, sorry, we're not interested. It's a nice story and so on. And we know that it was very limited in scope because it's very, well, it is quantitative, but it's not quantitative unless you really go after the data. And finding the data was quite difficult. So we did spend quite a long time now to trying to see how we can match that the best way to existing data. And this is, so now we have put a paper on bioarchive in the last few weeks on that. It's mostly the work of Pavan with a brilliant PhD student of mine, and a group of people in Lund, and the PI is Oscar Hansen, who have been doing this BioFinder database. So they've accumulated a lot of, they've been building a huge database over the years. And also people at Roche, on which we work, Roche also is very interested because they have drugs, possible drugs that they want to understand. We also look at another database, which is ADNIT. This is a public database, anybody can look at. It's a wonderful initiative where you have thousands and thousands of MRI and other data on Alzheimer's disease. It's one of the prime source for study. So what we knew also, and I told you already for like TPD43, there is a region that spares. And when you talk to people, they say, why is this region spared? Said, well, the level that this region of toxic protein that this region can take is different from one region to another one, or one patient to another one. And we knew from the get-go that our model was nice, but clearly it missed a huge component, which is local heterogeneity. That the transport process might be right, but that different regions have, from the data, we know different level of saturation, for instance. So we decided to transform that to a model with regional values. And the best regional data is given by the TOPET. The TOPET is standardized uptake value ratio. It's the value you get by imaging a radio tracer that binds to this tau protein. But SUVR is not a concentration, it's just the radio tracer, right? So you have to be careful. And so the model that we propose accordingly is a model where we know that you have a zero basis that is not zero, that when you look at the signal of the brain for this radio tracer, you always get a signal. Right? There is a healthy amount of off-target, it's called off-target binding. This radio tracer binds to the region, even if there is zero toxic protein, and that each region can have a different carrying capacity. So they can only have such a certain level of concentration of that, and that you have to extract from the data. And that's the only thing we did, which is what is the simplest possible alteration of the model that take into account this basic fact. So we have off-target binding, and we're going to see that's actually very homogeneous. And then you have this carrying capacity. So in order to do also model selection, you can compare different models. You can do the local model that I just showed you. You can go back to a global model. That's the first one I show you, that there are only 4 parameters. You say there is a global off-target binding and a global carrying capacity, and then you have alpha and rho parameters. You can do a pure diffusion process. We never believed that it was a good model, but that's the main model that's been used for the last 12 years to try to explain some of this data. So we wanted to say, well, we should include it to see how it compares. And a logistic model, which is what if there is no transport, that the property is purely local? So this is the same as the first model, but without the transport term. And that's also interesting because that's also something that people have been looking at, just analyzing data without trying to put a mechanism into it. So not surprisingly, diffusion is not very good because we know diffusion. And if you start with a certain quantity here, it goes down here, and you see that there is nothing happening in the brain. Right? It's not very interesting. No, the global one is the one I showed you before, and it reproduces some of the feature. It does the right staging and all that. But when you talk to people who know, like the people in Lund, they really know their stuff. They've looked at the data, they've looked at evolution and all that. They say, well, it's not quite right. This region shouldn't be attacked at that time. You know, it's not what we see. Now, the local one is more interesting. I'll show you a possible scan. You see that the local one, the SUVR, is not 0, right? But it's different here, and the value is different here also. And now you have something that's actually much, much closer to what is observed. So the question is, how do we get this value, this carrying capacity out of the data, right? So we have to infer these parameters. We have two types of parameter. We have the local parameter and we have the global parameter alpha and rho. And these are different. And so what you do, you use Gaussian mixture modeling. You say, well, in a given region, I can look at the SUVR signal and look at the distribution over many patients. And the many patients, we have 119 from ADNI that have at least 3 PET scan and 141 from Biofinder with at least 3. So we did the two separately without. And what you find is that when you look and you average and you look at one node, you see that you have this big peak, which is the healthy concentration. If you didn't have any disease, that's the only thing you would see. But as the disease takes place, you have an increase in concentration in other parts of it. So you have a distribution of concentration that's not. So you can look at these and then look at the offset. So you have the two parts. The first peak of the Gaussian give you the off-target binding for each region. And the second part, you look at 90 percentile, for instance. The second part gives you the carrying capacity. So that's the maximum value that you have at different region. And you see that each of these curves is a different region. So it's a different carrying capacity. Okay, and that sets the local heterogeneous value for the entire brain once you go that, you apply that for the population. Now, you can use Bayesian inference on the last two parameters. And so you can group the patient in different categories. And that's particularly important because they're known to have different type of mechanism. So the one that are Alzheimer, typical Alzheimer, are amyloid beta positive and tau positive. And that's the group in green at the top. And if you just look at this one, what you find is that you have a distribution of production here that's positive, and a transport rate that's also positive. And so you have alpha is much bigger than rho. So it's exactly like this front propagation problem that you had. Now, if I look at amyloid beta only, you know, I only get the healthy part of the tau signal. And the healthy part tells me that the production rate is negative. So it's very stable. It goes back to a state. Okay, so it's also what you expect. And if you go amyloid beta positive, but tau P negative, you also have average of 0 production. Okay, so that's all consistent. But let's focus on the green one. And what's remarkable, if you look at ADNI database with a given radio tracer, and you do the exact same procedure on the Biofinder 2 data with a different radio tracer, different protocol and all that, what you get is almost exactly the same value, right? If you look here, if you look at the green one, it's exactly the same value for the transport. So that really gives you confidence in your inference. You take 2 independent data sets, obtained in different methods with different chemicals, and you try to match your model, and you essentially find exactly the same value for both. But you can go more. You can look at the prediction versus the data, and you see it all looks good. The local FPP, they all, if the prediction was perfect, it all be unaligned. See, diffusion doesn't look that good, but the other one says. But since you have scanned, if you have longitudinal study, you can look at the evolution from one scan to the other one, and you can compare. That's a much finer metric. And what you see is that the diffusion is terrible, right? Because of course it goes down, so you really don't capture anything. The global one also is terrible. You really don't capture the correct change in one scan to the next one. However, the local FKPP is quite good, and the logistic is very good. The two are good. Now, if you do some metric of who's better than the other one, the local FKPP actually is slightly better than one, the logistic. And so you would prefer, in terms of model selection, you would prefer the local FKPP. But it's very interesting that the logistic one is so good, because it tells you that by the time T-scan are done, the brain is probably sufficiently seeded so that most of the fitting is about this logistic progression to different levels. Also note that since the carrying capacity changes, it really changed the linear part of the operator. So it's also changed the rate at which you increase in production. It's not just so much the asymptotic value. If you take a sigmoid and move it up, that means the middle part that goes up goes faster, the rate of change. You increase the slope. So that's what is also captured by this. Now you can play the game where you remove 10 patients that have 5 scans and consider what you would get from not training your data on it. You retrain the data without this, and you realize that individual prediction, each of these points is one node for one individual, is actually very good. The regional prediction, if you average that over the different patients, is also very good. So you can really go now to individual prediction, and you can, for instance, look at patient with 5 scans, and you take the 3 first scans to train the data, and you say, well, what would be the 4th and 5th scan? And the 4th and 5th scan would be within that region, 95 percentile, and you see that the square is where it is. And I took these as, actually, these are the worst prediction that you have from the set. This is, oh, sorry, let's go back. And you see that if you look not by region, but the entire brain, the prediction, that width is extremely small and very good. T-square really fall onto the average. So it tells you 2 things. It tells you that you can do regional prediction of the level. If you give me 3 PET scan, I can give you a very sharp window at the regional level. And you can do at the individual level also. You can do patient-specific and region-specific with this type of method. Of course, having 3 PET scan this is for research. PET scan extremely expensive. It's not like anything that go and take 3. But it's that, in principle, the model is sufficiently good to do this, so that it tells you something more about the model than the ability to do this prediction. Okay, so how am I doing with time? Oh, almost done. Okay, so just to wrap it up, we started with observation of the clinical data, and I show you that the continual model really captures something, and that the discrete model that you have also is a good match. You can match it against the continuum model and have it's a good proxy for the clinical data too. So I think we have now a good understanding of how to capture the spatial-temporal evolution, the atrophy related. We understand the biomarker I haven't told you about, but we can do much more about how these curves place against each other. We understand the evolution of the topet data, and there is another part, which is how to couple that to the functional part of the brain, the cognitive part that I haven't told you about. We've done also other work. I told you that there are 2 proteins, and it's no surprise that these 2 proteins interact with each other. The presence of amyloid beta rapidly increased the rate of tau protein, and that's an important mechanism in trying to understand also the dynamics. So we have model where we we couple these two types of proteins. They express themselves. They have different spatial-temporal patterns, and so the dynamics of this coupling process is very intricate and very important, because the accumulation of amyloid beta and tau protein is the one that is most related to atrophy and damage, but amyloid beta and tau protein influence the neuronal activity in a different way. It changed the way the cells work. So knowing both fields is very important for that. We're looking at data related to clearance, local clearance. We're looking at the damage done at the microvasculature and how it changed. And we have different variation of the model to try to understand therapy. And I'm going to stop there, unless you want to know about branding. Thank you.
Yes.
That's the one go over time.
So thank you very much for this inspiring talk. Is there any question or comment also from home? Atio, please.
So first of all, thank you very much. Two questions. One is more local, and the other one is a bit more exotic. So the local one is about understanding the model, both the continuous and the discrete cell. You have shown that the model, in spite of its simplicity, say mathematical simplicity, is capable of matching the real life evolution, which is somehow fantastic and also somehow surprising, right? So my question is about the Dij, the coefficient that you put in the what you call transport is and the fuel term. So this is a time-dependent problem. So this Dij is taken at the beginning of the story, or it doesn't depend on time, because your model apparently is not coupled to the mechanics of the contraction.
Yeah.
So I presume that the way to embed this information into your model is through the Dij, which should then take a longitudinal.
Yeah.
Is that correct?
Yeah, so in principle, you'd say, well, transport is going to change as you have atrophy, you change the distance, you change the number of connection and all that. And you can test that idea. So there are two things you can do. You can go and talk to people and say, is that probably important? Or you can actually test the idea, which of course I didn't talk to people. So we should talk more to people. So the first thing you say, okay, now I introduce this coupling and I see that whether it changed, and barely it has almost no effect, and I can understand why. And then you go talk to people and say, yeah, of course, we know that the damage that's done at the axon level of the white matter is very late in the process. So if you look at that, you can understand mathematically what's going on. If you have sufficiently high level of seeding, transport becomes irrelevant. And that was this model selection show you. The logistic without any transport, remove the transport, was you part of the disease, it just goes. And the fact that you can exchange toxic protein becomes irrelevant, actually. But yeah, in principle, you can. And if you do it, you don't see any difference.
So this is the more surprising, that's related by the second question. So even the logistic model is capable of catching the microscopic behavior. The very few times that we talk to doctors, they seem to know, at least Italian doctors that you've been consulting, they seem to know very little. They seem to be very almost unable to actually explain what's going on. And here we have a very simple model that is capable of producing the relevant information. Is it because perhaps I misunderstood what they mean is they cannot understand the functionality rather than the mechanical progression?
Well, it is a very complicated disease, and we take it in a place that's the doctors don't see. You know, we took this data, it's a big research database, and we extract information from that, and we see that it matched this simple ID. But on a patient level, what you see is the ends point of a very complicated multi-step process. You know, you have the toxic protein, and then you have atrophy, you have change in glutamate level that change the activity of excitatory inhibitory neurons that all of a sudden change the local biochemistry. And that's not even what they see. What they see is the way the patient behaves. And the patient changes behavior on a daily basis because one of the effect of hyperactivity is, for instance, lack of sleep, and that affects multiple processes. So they see the patient at the end of an extremely mixed and chaotic process that started with a relatively simple biophysical process at the end point. And we're not quite, I mean, I think we have a better understanding now, and a multiple group trying to apply our idea of coupling the toxicity to brain activity, but that's just brain activity. That's just, you know, the waves and so on. You still have quite a way to say, if I know that there is hyperactivity in the part of the brain, if I know some part of the brain is damaged, how does that actually change cognitively? If I know that a specific subnetwork, you know, there are different networks that are associated, motor network and all that, rest state and all that, default mode network. If I know these are affected, can I actually make a statement about the behavior or cognitive ability? And you see, you really go at the end, top of the pile, right? Because cognitive ability is all the way to, you know, consciousness and sentience and intelligence and relating multiple things around you. So they're trying to look at the top and say, well, this patient, you know, it goes to apathy, loss of consciousness, bouts of teeth and that. But this is so far, you know, so far above our ability to understand even in the healthy case. Now, what I find fascinating is that, and that's also why I started doing that. I said, well, you know, particle physicists, if they want to understand elementary particle, they go and smash the particle and see what comes up. So these diseases are really breaking a normal system and give us a window into how normal sentience, normal consciousness actually work. And I think that aspect is also interesting. But I think the basic aspect is that by the end effects are so complicated, so complex, the result of so much processes, that it does appear extremely complicated. And rightly so. We are complicated.
Yes, please.
Thank you for the talk. A couple of questions. At one point, you were mentioning that you're estimating the Alzheimer parameter of the model, and you were mentioning that regardless the type of images that you were using and the type of the tracer that you were using, the estimation was basically right. So I find this thing very curious because usually radio tracers, they are different, they target different physiological aspects of the biology and the process. So the question is, if you know which tracer you were using, and how can you like justify this finding?
Yeah, so I never remember the name. They are fluorobaxy something, but yeah, for the so there are two different ones. I can, it's in the paper. I can, these names are too complicated for me. But they do both target the tau protein. But I don't know exactly if they target the same oligomer to be. So I don't think it matters at our level because it's a kind of it gives us an average property. It's the same imaging technique, but on different type of scan, or the procedure that they use is different. So that's the first thing. The other thing is that, like every kind of data, you just don't take the data and just plot it and put it there. There's a lot of knowledge that's accumulated by people who work in these. And so the first thing that you do with the SUVR is that you have to standardize, normalize it, and you normalize it by the cerebellum region. Is that what? So in order to have that, and then there are regions that are known to not work with. There's too much white matter that are not good, that you also have to exclude, that are really outliers, and that you also have to do.
So it's the same with the tracer, but different like scanners.
Well, the actual radio tracer is different. Yeah, the actual radio tracer is different. The protocol of imaging is different, but the scanner typically are the same.
And the second question is maybe I missed it, but which is the data that you or anything that you use to validate the output of your simulations? I mean, how do you can say that they are like what's actually happening?
So yeah, so yeah, I mean, what kind of data would I need to validate? Yeah. I mean, the data you can validate the model by itself by removing some part of the data and trying to predict it. That's what I said. The out of sample. You say, assume for sample, I don't have this data. I'm hiding this data. You train your model. You have all the parameter, and then you say, okay, if I know the first 3 data point on this, can I predict the next 2? So that's out of sample fit, and that works very well. And so you have some metrics that actually tells you whether or not it's a good fit for that. So you can validate the model on the data that you already have, but of course you'd like more data, more longitudinal data. There's huge progress. A couple of years ago, we tried to do that with Alain in Stanford. And that release of ADNI did not quite, they had like 60 and all that. And we could only do that at population level, not at regional level, and not as patient specific, because the data was not good enough in terms of the number of patients with at least 3 scans. So you really depend. I think we're right at the border where we can do that. But the good thing is that this data set keeps increasing. I mean, the problem is not going away. So we get better data with years. And I think that's also exciting because you really realize that, you really depend on the best and latest data that you can find. And I would have much less confidence in the result if we hadn't done exactly the same procedure on 2 completely independent data sets.
Thank you again for the fantastic lecture. My curiosity is about vasculature. What's the role of vasculature in terms of the anatomy and also in terms of function? For example, does it have a role in the brain waves? So yeah, so at the physical level, vasculature is very small in the brain. It's only 3% of the total volume, but it's of course fundamental. It's the most irrigated or the most provided part of any tissue. Any kind of brain activity depends on the ability of the vasculature to deliver oxygen at the right place. And so a big part of the brain is the regulation of the microvasculature opening at the level of different places in the brain in order to release oxygen so that the axon can fire. A big part of a brain, of course, is of our energy expenditures to the brain. So vasculature is fundamental at all levels in brain function. It's a highly vascularized, very highly controlled system. It's just like university administration. The brain is the boss, so they get first all the resources and then give the resource to the rest. And in dire time, they keep the resource and the other ones have to cut from. The same for the brain. Brain maintain its blood flow as much as it can, even when the rest of the body needs it. It's the boss. It's the admin, right? And so it's extremely well regulated brain, which is that by itself as a control mechanism is also fascinating. So yeah, it depends at all levels. Now, there are other types of dementia, which is called vasculature dementia, that are really related to disease that affect directly the brain vasculature. And that essentially give the same type of mechanism, but are not necessarily driven by protein. At the end stage, they all look a little like Alzheimer when the damage is sufficiently large, but so they're important. Another part which we're looking at is we have no model about damage of microvasculature. And the idea is that if we know that the amyloid beta concentration is sufficiently high, then you look at the network, the microvascular network, and you damage it by removing edges, and you see how it changes conductance in time. And so you can, it's nice statistical physics there that you can do to see an evolution. And so when that happens, it further enhances the ability to create more of this type of defect. So it has also an autocatalytic effect. And so we've been looking at the role of microvasculature damage into the brain. At some point, I thought that the vasculature itself would be important. You know, if you have a damage in one region, you might get less blood, and then also have that. But it turns out that there are so many redundant mechanisms that's probably not the case. But the microvasculature, we think, play an important role.
Any further questions? Yes, please.
Thank you. Question, actually two questions. One is related to the logistic model. It was really surprising to me to see that almost good results at the end of this. Because in the first part of the talk, I understood that diffusion, particularly among microbial directions, so the transport along directions were the key features of this. So why in the logistic model you don't have that? Yeah. So I'm not able to.
Yeah, it seems a little paradoxical, and of course it was, you know, a little bit. We wanted our old model to be the best by far, but you can actually very easily understand that, and it's really the basic idea that the transport part is really doing this initial seeding, and that depends very clearly on the topology of it. But once you have this seeding, you know, you start increasing at different rate at different place, and that's what you see. And by the time this scan were taken, there was already far enough in the disease progression. So it's not in the flat part, you know, and that's good for us because you really want the data when things change. So you're already a stage of the disease that advanced so that the actual transport part become less important. You've already set the scene. You put all your actors there, and then they start moving differently. With some delay, that's due to the transport. So we still think it's important, that the transport process, but it's like a two-stage process. You first transport and move, and then you go. And amyloid beta is even more striking. I think amyloid beta is more or less diffuse everywhere, and then it's only local properties that makes it grow. And we think that the presence of tau is closely related to amyloid beta. When you have this level of amyloid beta, then tau is going to start increasing also.
Hi, can you hear me?
Related to the misfolding,
No.
just mentioned that normal proteins do not stick together, while misfolded proteins do stick together. Mm-hmm. So is there any mechanical reason for that?
Yeah, I mean, well, not so much in the tau. Tau is quite complicated. There are a lot of models for amyloid beta fibrils, amyloid beta plaque formation. Tau is interesting because tau has essentially heads. It has a side that binds to the microtubule and another one, and its function is to bring it together and glue the two parts so that they maintain the integrity of the microtubule. So you can think of it like a little zipper. Now, the way this zipper function is that they have a lot of phosphorylation sites, and it's believed that the misfolding comes from the fact that when you have hyperphosphorylation, some of the parts can fold on themselves and then create a structure that's more likely to have multiple of these proteins to start sticking together. But there is a whole aspect of change or configuration. There are different, what's called isoforms of tau, so different taus that, you know, if you look in different mammals and all that, they're slightly different. They have different sequence. But now there is a group in Cambridge who is very well known for that, to do conformation of that, extract the conformation. I don't know any good spatial, like physical models of that process. It's very, very difficult to get that, the dynamics of it.
But that would be very interesting also.
Sorry.
Okay, if there are no more questions, let's thank Professor Goriely again for the great talk. We do have the coffee break at 6th floor in near the entrance, the MOX entrance.